Zika virus and vaccine. Zika virus, ZIKV, is a mosquito-borne flavivirus belonging to the Flaviviridae family. It is closely related to other viruses such as Dengue virus, yellow fever virus, West Nile virus, and Japanese encephalitis virus. Origin and history. Discovered, in 1947, in a rhesus monkey in the Zika forest of Uganda, hence the name Zika. First human case, detected in 1952 in Uganda and Tanzania. Major outbreaks. 2007, Yap Island, Micronesia, first major human outbreak. 2013 to 2014, French Polynesia, widespread infection. 2015 to 2016, Brazil, associated with microcephaly and Guillain-Barré syndrome, GBS, marking it as a major public health emergency declared by WHO, 2016. Structure of Zika virus. Feature. Description. Type. Enveloped, icosahedral virus. Genome. Single-stranded, positive sense RNA, 10.7 kb. Family. Flaviviridae. Genus. Flavivirus. Proteins. 3 structural proteins and 7 non-structural proteins. Structural proteins. C. Capsid, PRM/M, membrane, E. Envelope. Envelope, E. Protein. Major surface glycoprotein responsible for host cell binding and fusion. Replication site. Cytoplasm of host cell. Transmission. Primarily through Aedes aegypti mosquitoes, also, via sexual contact, blood transfusion, and mother to fetus, vertical transmission. Pathogenesis, mechanism of disease. 1. Transmission and entry. Aedes mosquitoes bite and inject the virus into human skin or bloodstream. Virus binds to host cell receptors, example DC sign, AXL, Tyro3, on skin fibroblasts, keratinocytes, and dendritic cells. Enters via receptor-mediated endocytosis. 2. Replication. After entry, viral RNA is released into the cytoplasm. Translated into a single polyprotein, cleaved into structural and non-structural proteins. Replication complex forms on the endoplasmic reticulum, where new viral RNA and particles are assembled. 3. Cell damage and spread. ZIKV causes apoptosis and autophagy in infected cells. Infects neuronal progenitor cells in fetuses rightward arrow microcephaly. Can infect immune cells, testicular tissue, and placental cells, leading to sexual and vertical transmission. 4. Immune response. Host immune system activates type I interferons, NK cells, and neutralizing antibodies. Cross-reactivity with dengue virus antibodies can lead to antibody-dependent enhancement, AID, increasing infection severity. Why Zika is an emerging disease. Geographical expansion. Spread from Africa to Asia to Pacific Islands to Americas. Multiple transmission routes. Vector-borne, sexual, and vertical transmission increase its spread. Severe complications. Linked to congenital Zika syndrome, CZS, microcephaly and brain malformations in newborns. Neurological disorders, example Guillain-Barré syndrome, in adults. Lack of approved vaccine or antiviral drug. No specific treatment or vaccine available yet. Global travel and urbanization. Increased mosquito breeding and global spread. Need for a vaccine. To prevent congenital defects caused by maternal infection. To control epidemics and reduce morbidity. To protect travelers and endemic populations. To avoid antibody cross-reactivity and aid from co-circulating flaviviruses. Antigenic components of Zika virus for vaccine development. The Zika virus, ZIKV, genome encodes a single polyprotein, which is cleaved into 3 structural proteins rightward arrow form the virus particle, virion. 7 non-structural, NS, proteins rightward arrow involved in replication, assembly, and immune evasion. Envelope, E, protein, major antigen, largest surface glycoprotein, involved in host cell receptor binding, membrane fusion, and viral entry main target for neutralizing antibodies induces protective humoral immunity. Premembrane/membrane, PRM/M, protein, works with E protein during virus assembly and maturation. PRM stabilizes the E protein conformation in immature virions and is cleaved to M protein in mature virions. Capsid, C, protein, forms the nucleocapsid around the viral RNA, contains T cell epitopes that can activate CD8 cytotoxic T cells. Non-structural, NS, proteins as antigens, although NS proteins are not part of the viral particle, they play critical roles in replication and immune modulation and can be powerful T cell antigens, NS1, NS3, NS5 etc. Strategies for designing effective Zika vaccines. Several platforms are under development or in trials. Vaccine type. Approach. Mechanism and notes. Example/stage. 1. DNA vaccines. Plasmid encoding ZIKV-PRME proteins. Host cells express viral proteins. Rightward arrow elicit T cell and antibody response. NIAID DNA vaccine, VRC5288, VRC5283, Phase 2. 2 mRNA vaccines. mRNA encoding E and PRM proteins. Direct translation in host. Rightward arrow rapid immune response. Moderna mRNA 1893, Phase 2, CureVac. 3 inactivated virus vaccines. Chemically inactivated whole virus. Safe, stable, elicits neutralizing antibodies. Bharat Biotech, Zika Vac registered, preclinical, Walter Reed Army Institute. 4 live attenuated vaccines. Weakened ZIKV strains. Strong immune response but risk in pregnancy. ZIKV lab, attenuated via NS1 mutation. 5 viral vector vaccines. Adenovirus, measles, or vaccinia vectors expressing ZIKV antigens. Induce strong cellular and humoral immunity. Ad26.ZIKV, Janssen, MVZIKA, Themis Bioscience. 6 subunit vaccines. Recombinant E or EDIII proteins. Safe and specific, but may need adjuvants. ZIKV-E protein nanoparticles. 7 virus-like particles, VLPs. Non-infectious particles mimicking virus. Strong antibody response without risk of infection. VLP-ZIKV candidate, preclinical. Ideal Zika vaccine antigen should induce strong neutralizing antibodies against E protein. Activate T cell responses against C, NS3, NS5. Avoid cross-reactivity with dengue and other flaviviruses to prevent Aedes. Be safe for pregnant women. Be thermostable and easy to produce. Challenges in vaccine development. Cross-reactivity with dengue and other flaviviruses. Risk of Aedes. Need for safe vaccines for pregnant women. Requirement for long-lasting immunity. Limited animal models that mimic human fetal effects. Future directions. Structure-based antigen design, targeting specific neutralizing epitopes on E protein. Multivalent flavivirus vaccines, covering dengue plus Zika. Nanoparticle or mRNA-based platforms for rapid scalability. Passive immunization using monoclonal antibodies for high-risk groups. Zika virus, though once obscure, became a global health concern due to its neurological and congenital complications. Developing a safe, effective, and pregnancy-compatible vaccine remains a top global health priority, especially in tropical and subtropical regions. Thanks for watching.